Today I'm going to be talking about the evolution of a high-throughput environmental DNA sampling platform that we've been developing here at Smith Root. My name is Austen Thomas, and I'm a senior research scientist here, and I've been overseeing the group that's been developing it. Smith Root is a small fisheries conservation technology company based out of Vancouver, Washington, USA. I'd also like to acknowledge my co-author on this talk, Caren Goldberg, with Washington State University, who's been integral to the development of this platform. So in 2015, we looked around at kind of the available tools for eDNA sampling and found they had a set of limitations. Mainly that the pore sizes of materials available were very limited and mostly designed for microbial studies. This limits the sample water volume and therefore the detectability of many species. And there's also minimal control over the filtration process. These tend to be very simple DC pumps or syringe filters. And the sampling gear is not purpose-built, so it was pretty cumbersome. So we thought, what can we do to help improve this process? And this is what we came up with. So the main components of the sampler are these. So it's a computer-controlled pump system for rapid eDNA filtration, and it has a remote control. Custom eDNA filter packets with the potential to put different pore sizes and membranes in those filter packets. Also on board this computer-controlled pump is a GPS, pressure and flow sensors, and with programmable thresholds, and it generates a complete metadata record of each sample. The other really cool thing about having a backpack-mounted filtration system is that you can do mobile sampling. So our thought in the design was to make a system that would facilitate mobile sampling when that becomes a more common part of eDNA protocols. The idea being, of course, that eDNA is heterogeneous in space, and mobile sampling, a spatially integrated sample, might do a better job of representing the DNA in the environment. So you can easily invert those filters, move around obstructions, etc. And you can do multiple filters simultaneously, and using the flow rate control, stretch out your sampling over space. So we were feeling a little bit bad about having developed a system that produces a fair amount of plastic waste, and so we started looking into alternative types of plastics that are biodegradable and discovered that they're actually, many of them, highly hydrophilic, and that got us to thinking, hey, we could actually make a self-preserving filter if it automatically desiccates that membrane. So the advantages of a self-preserving filter is that there's no membrane transfer step, which reduces the chance of potential contamination, no chemical or cold storage required in the field, and then that reduces your overall field time. And we found, and we also published this as well, that the eDNA is stable in these self-preserving filters for up to 6 months. And I'm just going to go ahead and show a quick video of what that sampling process looks like. So you'd open up the filter packet, pull out the filter by grabbing just the barbed part of the filter housing and attach that little snorkel extension tube, attach your suction tubing, hit go on the pump. It will automatically prime itself, and then filtration continues until you get that double beep indicating that you've hit your target volume. Allow the pump to run to air dry that membrane as much as possible so that biodegradable desiccating plastic doesn't have to work too hard. Seal it back up, stop the pump, remove both sections of tubing, and just put it back in the bag. And then the next steps for this system, so we've found that not everybody likes the backpack model, and some people really like the computer-controlled pump, but they'd like something a little bit more compact, so we created a handheld model for it. We're working on a visualization and data management software so you can look at a bunch of different filtration events simultaneously and potentially identify outliers in those events. We're also working on, and should have available this coming summer, a lower cost vacuum pump model for citizen science projects. And combined with those self-preserving filters, we think will make the sampling process very easy and fast for higher volume samples than what can be done with syringe filters. And we also have an ongoing study working with WSU looking at mobile sampling by comparison to point sampling. And what we're finding preliminarily is that it does improve the overall eDNA capture for certain target species, particularly when you move and target certain environments through a particular transect. And I'd love to take people's questions. I know there's not a whole lot of time, but thank you very much for listening, and I look forward to the rest of the talks. Thank you.